a big advantage. Yes, so, and I would like, as this is always tradition in this advanced mass spectrometry seminar, I would like to give this educational teaser now, which is more like a lecture type of style, where we would like to introduce typically the technologies that those speakers that present later on were using in their research. Yeah, but before I do that, I actually first would like to give you a general overview over all the different types of how data can be acquired in LC-MS, MS, or liquid chromatography tandem mass spectrometry, no matter whether we're talking about metabolomics or proteomics here. Because I think in general, you can say that all these different data acquisition methods that are out there, they can be distinguished in two big groups. We have the targeted mass spectrometric methods, where you really just acquire data for those analytes, whether it's metabolites or peptides, proteins that are of interest for you. And then there is the field of untargeted, or some people call it discovery-driven mass spectrometry, or hypothesis generating mass spectrometry, where the aim is really always to acquire the data on the mass spectrometers in a way that you would like to ideally detect everything that is in the sample. So all peptides or all metabolites, or maybe both of them even at the same time. So if we come now to different data acquisition methods for targeted mass spectrometry, there are two different types that are currently used in the field. One is called selected reaction monitoring, SRM, and the other is called parallel reaction monitoring. And I will explain you what's the difference. And when looking at untargeted or discovery-driven mass spectrometry, we have also two different ways of how to operate the mass spectrometer. And those are called data-dependent acquisition methods or data-independent acquisition methods. Now, as this title slide already suggests, unfortunately, in the field of mass spectrometry, there are not just these four different types of techniques, but there are a lot of more flavors and a lot of more different nomenclatures than the four names that I showed you here. And that makes this whole thing often a bit complicated. So for example, for the data acquisition method SRM, there's also the term MRM, multiple reaction monitoring, around. And the reason why for this additional name is trademarking. So the company SciEx trademarked this term MRM to their mass spectrometers. So if you perform an SRM experiment on a SciEx instrument, you should call it MRM, multiple reaction monitoring. But SRM and MRM are really the exact same thing. So the way how the mass spectrometer operates in SRM or MRM, there is no difference. It's just the name of the vendor that you use. The same holds true for parallel reaction monitoring, PRM. So there is also a trademark term from SciEx, which is called MRM-HR, so MRM high resolution, or sometimes you also find in the literature pseudo-SRM or pseudo-MRM. And then for data-dependent acquisition, back in the field of untargeted mass spectrometry, SciEx again also trademarked the term IDA, information-dependent acquisition. So sometimes you read IDA instead of DDA. Or in the field of proteomics, also many people speak about shotgun proteomics, and then they also usually mean this data-dependent or IDA, information-dependent acquisition proteomics. And the field of DIA now is even the most complicated of all, because in DIA, which is also historically speaking the most recently developed field, or where a lot of active research is ongoing, and that's the field with the biggest variety of different technologies. So yeah, there's a huge array or very different flavors of how you can generate mass spectrometric data in DIA mode. And on top, there are also different nomenclatures and different trademarks. So for example, a very popular DIA acquisition method in mass spectrometry is SWOF-MS. Maybe you've heard about it. It's again a term that is trademarked by SciEx. There's also the term HRM, hyperreaction monitoring, trademarked by a company called Biognosis. And also different vendors have slightly different ways of acquiring DIA data using different parameters or using different analysis strategies called Sonar, which is the Waters company, or MSE or BIPASF. So there are all differences in the way how bigger or smaller differences in the way how the data gets acquired, but they all fall under this big umbrella of DIA. And there can be, I can talk a long time about all these differences in different acquisition methods in DIA. Okay, so I hope that this overview of all the different terms and nomenclatures and trademarks will maybe help, especially people who are new in the field, to get an idea of where are really things different and where same names actually refer to the same technology. Now, our two speakers of today, Vlad and Sven, they both performed measurements here in this world of targeted mass spectrometry. And Vlad did, his measurements were performed on a triple quadruple instrument from SciEx. So he did multiple reaction monitoring, not SRM. And Sven, on the other hand, who performed targeted proteomics measurements, worked on an Orbitrap mass spectrometer, and he did parallel reaction monitoring. And on the next slides, I would now quickly like to once more introduce these two different ways of acquiring data, and also once more summarize what are maybe the advantages or disadvantages of these technologies. So let's get started with SRM or MRM. How does that work? So SRM and MRM measurements are always performed on so-called triple quadruple mass spectrometers. And as this name already suggests, these mass spectrometers consist out of 3 quadruple mass analyzers that are used in a row. And the first and the third quadruple both serve as mass filters. So what does that mean? So if you have a mixture of metabolites or a mixture of peptides eluting from your LC system at a certain time point, then these mixtures of analytes will enter the triple quadruple, and the first quadruple then will just isolate one of these analytes out of the mixture. So in my little cartoon here, the green ball, it will then fragment this analyte into its fragments in the collision cell, which is the second quadruple. And the third quadruple then will again filter just one of these fragments out of the mixture, and the detector here at the end will then detect the intensity of this fragment ion. So what you do in SRM or MRM mode is you really always isolate pairs of precursors and corresponding fragment ions. And the precursor you isolate in the first quadruple, and the fragment ion you isolate in the third quadruple. And the scientific expression that is used in the field for this Q1, Q3 precursor fragment ion pairs is called a transition. So you record intensities of transitions, and you never record at no time point a full spectrum. So you never record all the analytes that elute from your column, and you never record all fragment ions. You just record intensities of selected transitions, but those ones you record consistently over time, and then you can generate these extracted ion chromatograms, and you can use those areas under the curve for quantification. However, before you can start press the start button on a triple quad to do this type of measurement, what you need is what is called a transition list. That means you need to tell the mass spectrometer the exact masses, M/Z values that it should isolate here in Q1 and Q3. And ideally, you also need retention time information to these masses. So this type of table where you tell the instrument this M/Z value and this fragment of this precursor, this is what I would like to analyze. So this needs to be available before you can do such a measurement. What are the advantages of using triple quads and doing such SRM, MRM measurements? Now, triple quadruple instruments are very efficient in reducing noise and background intensities. And this is due to these two filtering steps here. So both quadruples are two ortholog filters, if you like. And that's why you can very efficiently reduce all the other signals that are in your sample as well. Quadruples have a high ion transmission efficiency, so 95% of the green ball will actually make it through the quadruple. And you can also rapidly switch between different transitions, so you can not just record one precursor fragment ion pair, but you can also record in a single measurement many fragment ion pairs of several fragment ions of the same analyte, or several different fragment ions of the same analyte or also many different analytes. So triple quads are very sensitive and reproducible and accurate. But the big disadvantage of triple quads is the low resolution. So especially compared to TOF mass analyzers or Orbitraps, the resolution of a quadrupole is really very low. And that means that the selectivity that you can achieve, so how accurate you can measure a specific mass, is really very limited. And you have the disadvantage that you need this transition list. So you need to know what to look for in advance. And this transition list cannot be endlessly long. So in proteomics, we often say, yeah, we can measure maybe 10 to a few hundred peptides in a single measurement, but that's the absolute maximum. So you cannot do this measurement for a large number of analytes. Okay, so then let's switch to PRM or MRM-HR mode. How does a mass spectrometer function when you do this type of measurement? And PRM or MRM-HR measurements are not performed on triple quads, but they are performed on quadrupole Orbitrap or quadrupole TOF mass analyzers these days. And here the mass spectrometer functions in two different scan modes. So the first one is always an MS1 full scan. So what the mass spectrometer does here is, again, we have our mixture of analytes eluting from the LC. The quadrupole will be switched off. So all analytes will just pass through. And the high mass accuracy, high resolution Orbi or TOF mass analyzer will simply record all the analytes that are eluting at that time point. And then the instrument will switch to MS2 scan mode. And what that means is that now the quadrupole gets active, and it will, just as we saw it for SRM and MRM as well, it will isolate specific balls, specific analytes, will fragment them and record the complete fragment ion spectrum of these analytes. And this is not just done for one precursor, or for one peptide or one metabolite, but usually this is done for several peptides in a row. And after a few seconds, typically, then the instrument switches back to the full MS1 scan, and the cycle of MS1, many MS2s, is usually performed over the whole LC gradient that you apply and the whole MS run that you perform. So what's very important about this PRM run is that these MS2 scans that you do here are completely independent from the MS1 scan that is performed up here. So it doesn't matter which signals are detected here in MS1, but instead what you do is you again provide the information, the user, the operator of the mass spectrometer needs to provide the information, what should be the analytes that I would like to isolate here in all these MS2 scans. So you need to provide what is called a precursor target list. So you need to provide the precursor M/Z value and the retention time. However, in PRM or MRM-HR mode, you do not need to provide also fragment ion information because the instrument will always acquire the complete MS2 spectrum. So only precursor information is needed. However, when you acquire data in that way, when you then want to analyze it, then you can use this data and also generate extracted ion chromatograms, just as I've shown it to you before for SRM measurements. And in order to do this, you actually do again also need a transition list because you need to take this data and now extract again specific transitions from all of these spectra. And then also fragment ion information is again required, but it's only required at the stage of data analysis, and it's not required at the stage where you acquire the data. Okay, so this is my last slide, conclusion. Now, what are the differences between PRM and SRM, and when is maybe what is advantage and disadvantage of each of them? So the advantage of this PRM or MRM-HR method that I just showed you in comparison to SRM is the high resolution and the high mass accuracy of all these MS1 and MS2 scans that you acquire. So that gives you a better selectivity of the information of the measurement. And you have this full MS2 scan information. So you get in this measurement always all fragment ions that are formed by a peptide. And that means that if you want to, for example, remove a certain fragment ion because it's not maybe interfered or not performing well, you do not need to reacquire the data, but you can just go back into the data and extract another fragment ion from the full MS2 scan that you have. And this is something you cannot do in SRM, where if you want to change certain transitions, you need to really reacquire the data again. And this is also the reason, so you just need a precursor isolationist, you do not need any fragment ions, and that is why the method setup to do a PRM measurement is a bit less work intensive than the method setup when you do an SRM measurement. However, disadvantage of PRM compared to SRM is speed. So it usually takes longer to acquire these full scans than to just acquire transitions. And also these, compared to a quadrupole, an Orbitrap or an POF has this less effective ion transmission. Overall, people say, so what is better? And there are lots of papers out there that also compared PRM to SRM in terms of dynamic quantification range, or what is the more accurate method, or where can you measure more analytes. And maybe there are some papers who describe that one is a bit better or the other is a bit better. And overall, I think it's fair to say that both of these targeted methods actually perform pretty similar and can give you very similar results. There is not one clear winner over the other. So when does it then make sense to use the one or the other? So if it's now your turn and you want to do a targeted measurement, MS measurement, how should you choose? Which one should you select for? And here are just some points that I can just, that I came up with when it maybe makes sense to do PRM and not SRM. And the first argument to do a PRM measurement, measurement, and this is very often a big argument for me, myself, is that if you don't have a triple quadruple instrument available, then you cannot do an SRM or MRM experiment. Then you're only left with doing PRM. And for example, for proteomic measurements, I do not have a triple quadruple available. So I, in the facility, always perform PRM measurements. Also, the reason to do PRM and maybe not SRM or MRM is if you want to stay with your mass spectrometer on the same platform where you have also performed previous discovery-driven analysis. So very often projects function in a way that you first do a global discovery or untargeted analysis, and then you want to do some follow-ups on just specific interesting targets. And if you want to do this on exactly the same instrument, then this can also only be done in PRM mode, because only PRM is done on the same mass spectrometer as DDA or DIA. Also, if selectivity really matters, then PRM can be beneficial because of the higher mass accuracy and higher resolution of the data. It is possible for specific cases when you have analytes that are very, very similar in mass, so almost isobaric, but not completely. Then it can be that with PRM, you can actually distinguish them, while with SRM, they just fell under one peak. But that's a very specific application. And also another specific application for proteomics is PTM site localization. So if you're interested in really localizing at which amino acid a certain PTM sits, it might also be beneficial to use PRM, because then you just get always the full information of all fragment ions that are formed, and this sometimes can help you in localizing a PTM better than in SRM, where if you did not acquire the correct or the most interesting transition for defining the PTM site, this is sometimes a bit more difficult or needs a lot of prior investigation and knowledge about this PTM site. Okay. With this, I'm at the end of my introduction lecture. I think the stage is prepared now for our two speakers who use these technologies, but maybe there are some questions from the audience. If there are some urgent questions, I'm happy to answer them. Is there maybe something in the chat also? Doesn't look like.
So feel free to ask. So maybe I start. When I looked at the slide, I had this question why her second fragment ion is bigger than her precursor ion, which metabolomics people usually don't account. Maybe you can comment that for metabolomics people. Because the proteomics guys, they can use some beneficial stuff we cannot use to have more selectivity.
You mean this table here, right?
Yeah.
And that we have a bigger mass of the fragment than the precursor. Yeah, this is simply due to the fact that in proteomics, the peptides are usually doubly or triply charged. So the precursor is divided by charge 2 or 3. So the value is smaller, while fragment ions are typically singly charged. So, and that's why the fragment can be bigger than the mother mass. Actually, we like to use them because these fragment ions above the precursor mass are often very clean and have a low background noise and stuff like that. They are specifically good for quantification.
Okay, does anybody else have a question? We can also stop recording during the question Q&A sessions. Because later on you will find also those talks on our homepage if you just want to look up something. So please.
I don't want to start in the middle of the presentation. Okay, thank you for the introduction. Indeed, I am doing targeted neuropeptidomics in Caenorhabditis elegans. For the people that have never heard of what Caenorhabditis elegans is, I will do a short introduction about it as a model for neurobiology. So C. elegans is actually a small nematode, non-infective, which we can grow actually on agar plates, like you see in the picture here. And it feeds on bacteria, which is the big dot that you can see. It has been a popular model organism for neurobiology already from in the 80s when the entire connectome of its brain, well, brain, so C. elegans has 302 neurons, which are considered as a kind of mini brain, actually. So already in the 80s, this entire connectome of these 302 neurons was already published with more additional publications on this. This one is from 2019. Of course, this is not the entire connectome, but you can have an idea how much knowledge we already have on this brain. And it lends itself extremely well for all kinds of molecular biology techniques, such as, for instance, CRISPR nowadays. Lots of research groups are working on all kinds of things, like, for instance, the Sentient project had as a goal to make a complete gene expression map of the entire nervous system. And this is very recently, for instance, there's this Neuropeptide system, which is basically an atlas of the entire C. elegans neuronal system. So basically every neuron has a different color or a semi-different color. It basically functions as an atlas. You get these nice graphics, which makes it easier to actually identify neurons, what they're doing, etc. So for me, I'm a part. So in Leuven, we're actually with 3 labs that have that use the elegans for neurobiology. So these are the labs of Lilian Bates and Lisbeth Temmerman. I am part of the still am actually part of the Temmerman group at Leuven, and some of the keywords that you can see are all neurobiology related. And what they have, all these 3 labs have in common is neuropeptide. So lots of their research focuses on neuropeptides and how they regulate certain states of behavior with C. elegans as the model organism. So maybe a little explanation, what is a neuropeptide? Neuropeptide or neurohormone. I think many of you have heard of them. I have here some titles of publications which are more C. elegans centered, but I think many of you have heard from things like vasopressin or oxytocin, or thyroid releasing hormone, or gonadotropin releasing hormone. So neuropeptides are basically these kinds of peptide hormones. And as you can see from the publication, a lot of these have been conserved in C. elegans. So the mammalian versions are often present in C. elegans. So it's a very useful system to study neuropeptide systems. So this neuropeptide synthesis, this is actually very conserved around in the animal kingdom. So mainly neuropeptides are expressed as free propeptide, so it's a protein basically, a pro version of a protein, which is getting cleaved in sequential steps. So the propeptide gets the single peptide removed, which is necessary for targeting it to the endoplasmic reticulum. And you have then these maturation steps where different enzymes cleave these terminally of these dibasic sites. These get eventually removed, and some of the neuropeptides get additional modifications like this amidation or pyroglutamation. So actually, it's this set of mature peptides that we want to observe or want to detect with LCMS. So in C. elegans, you have a large repertoire of these neuropeptides. They have quite different names compared to what you're used to in invertebrates. For instance, they have two big groups, this FMRF amide neuropeptides, which are basically named because their C terminus contains the sequence FMRF or RF, and neuropeptide-like proteins. Basically, everything what is not an FMRF has been ended up in the group neuropeptide-like. So the names are actually pretty random. For instance, the oxytocin homolog of neuropeptide in C. elegans is actually something called NLP70 or something. So the vertebrate homologs are present in there, just the names are a bit different. And so in C. elegans, you have 400 of these mature neuropeptides, which is also quite a lot compared to what you're used to in vertebrates. So since this is mostly a proteomics public that I'm talking to, I think I want to stress or want to explain the difference between what is proteomics versus peptidomics, because they're very closely related, of course. They use the same, basically the same tools, but that doesn't mean that they're exactly the same in their setup. So I think everyone knows what a typical proteomics workflow looks like. You have your cells, tissue, organ of interest, you do a protein extraction and a digestion to get cryptic peptides that you put on the mass spec. And in your data analysis part, you puzzle it back together to get the complete protein again. For peptidomics, it's a bit different. So of course, we're looking at peptides, so we're trying to do a peptide extraction, which is basically we try to use solvents that precipitate as much of the protein as possible. So we end up with these biological peptides. So there's no digestion step necessary because these are biologically present. Why would we need to cleave them? And then actually, after some cleanup steps, we put them also on MOSPEC, so for LCMS. And the data analysis, well, what you have here is a bit of a representation what you expect to end up with, because of course these neuropeptides get cleaved from larger precursor proteins. You don't have the entire precursor puzzled back together. You only have the sequences, which are actually the neuropeptides. I want to stress also, it's not our goal to have, like in proteomics is the case, to rebuild the precursor, because the precursor is not what we're interested in. It's the peptides themselves that are our focus of interest. So you may ask, like, how do you know what's a neuropeptide? Because you have to just these random sequences within a larger precursor. So neuropeptide discovery is mainly based on two approaches. The first one is an in silico approach. So for instance, phylogeny, you just look at conservation of neuropeptide sequences. So for instance, the example that I gave, you can look for oxytocin-related sequences in other species and try to discover the right precursors. Or you can look at the proprotein characteristics. So proproteins that encode or contain the neuropeptides typically have some characteristics that are easily or can be recognized using certain in silico approaches. So for instance, they should contain this signal peptide, it should contain these biphasic sites that are cleavage sites to release the neuropeptides, amidation sites, and typically they're no longer than 300 amino acids. And of course, there's also just an LC-MS approach, which is mainly data-dependent acquisition. So discovery, you do your neuropeptide extraction, as I explained, you put it on the mouse spec and you see what you get out. Of course, these two approaches feed into each other because what you detect on your LC-MS, you can verify using the in silico approach and vice versa. So this was actually something that I did during my PhD research. So there are more than 400 mature neuropeptides that are both predicted and observed. So it's a combination of these both approaches. And currently in our lab, we have MS evidence for, say, elegans, at least 247 of these using DDA approaches. So having this, at the end of my PhD, we wondered how can we, for instance, overcome the variability between different DDA runs, because something that you typically see when you're doing DDA is you can have 10 samples, run them all, have something like 100 neuropeptides discovered in each sample, and if you then check how many are overlapping between all of your samples, you can then end up with 0 in the worst case. So there's quite a big variability if you run samples using a DDA approach. And how can you increase sensitivity? Because neuropeptides typically are not the most abundant peptides present in biological samples. So these were two main questions when we started to wanting to develop this start neuropeptidomics approach. So actually, we're in luck. This is the website of what's called the Peptide GPCR project, which is led by 2 people from Leuven, which I already mentioned. So it's a lab that two labs that we closely work together with. And actually, what they wanted to do is to deorganize G protein-coupled receptors in C. elegans. And what they had to do for this was to basically synthesize every known and predicted neuropeptide in C. elegans. So they have this library that they use for their deorphanization project, but it's also open to us to make, for instance, a targeted neuropeptidomics method with, because we have these synthetic versions of neuropeptides that we can measure on LC-MS and basically make a library with. So that is actually the first step of what I did with EPIC-XS at BayBioMS with Tina. Is we had these 427 C. elegans neuropeptides, and we added IRT peptides to for retention time calibration. And actually what we did was DDA runs to create a spectral library and then developed 2 methods. One was a scheduled PRM method, and the other one was actually also a data independent acquisition method, just for comparison's sake. So we grew worms in liquid cultures, so you can keep them on plates or grow them in liquid cultures, which is easy if we need large volumes of worms. And actually created 4 samples on which we did a neuropeptide extraction, and these 4 samples were split again. So just the biological extract and the biological extract spiked in with the synthetic library as a control. So yeah, like I said, we used these methods and we tested this on the extracts, and we did parallel reaction monitoring, a data independent acquisition, and a data dependent acquisition just for comparison. So everything was done in Skyline, and what I want to show you is, so this is a typical view that you get from Skyline. On top, you see the MS1 traces, so you have your monoisotopic peak and 2 isotope peaks. What you can see here is, for instance, for this ion, there's a very clean isotope peaks available. For this and this, not so much. If you then look at the MS2 traces, so these correspond to the ones on top, you can see that actually MS2, you have fairly nice peaks in there, which are all detectable and integratable, so you can do something quantitative on this. Of course, not all the data look like this, but I just picked these out to just show you what targeted methods can do. If you just select the right ions, you can have very clean MS2 ion fragments, while there's basically nothing to see in the MS1. So results. So we had our spectral library and a PRM inclusion list made from that, which contained these 497 entries for 288 neuropeptides. So we're about 2/3 of our known neuropeptidome. Of course, there are more entries than neuropeptides because we also included for some neuropeptide different charge states and different oxidation, so post-translational modification states like oxidation. So the results here, so how this should be interpreted is, so we compared PRM, DIA, and TDA. The black range gives the different number of identifications in the 4 samples. So for PRM, this was in the range of 172 to 178 neuropeptides that we could detect. And the red number is actually what was shared, the shared amount of neuropeptides in all samples. So in all 4 samples, in this case, we found 172. For DIA, it was a bit lower. So in all samples, we had 128. And DDA is what you expect is actually the number of overlap is lower than the minimum of one of the lowest samples. So just for comparison, so we have an 87% increase for DIA, and of course, PRM performs best of all, which is not a very big surprise. More than 140 identifications. So while DIA performs a bit less than PRM, it's still a very nice method, especially when you compare it to DDA, which we have been doing for the last 6 years. So even DIA is an for comparative and quantitation method is still very nice. So our control is the biological sample plus the spiked in peptides. This is just to test how well the method worked. So for PRM, yeah, 259, and for DIA, 220. So for PRM, basically, this is 90% of what is in the list. So this is very successful. DIA was a bit lower with 76%. Just for comparison's sake, we also compared the areas of the individual peptides. So what you see here are the 4 samples that we have run. And on the y-axis here is the fragment areas of the DIA method, and the fragment on the y-axis, sorry, this is the x-axis, of course. On the y-axis, the fragment area of the PRM method. You see that these are very linear, but not when they reach a certain intensity threshold. So when they the intensity or the area is lower than 10 to the 8, you can see that there's far more spread on the data, which is something that is known, of course. DIA is less sensitive than PRM, but it's interesting to see that is also the case in our data. So to round off, the purple, well, pinkish circle here shows what we have discovered using DDA methods. So this is a combined total of 31 runs that were done between 2014 and 2019. So these are the 245 neuropeptides that we have MS proof of. The yellow circle here is everything that was in our list. So that was based on our synthetic library that we measured. And the blue one here is actually what we have detected. What is interesting here is that there are actually 25 peptides that were measured using the detected methods that we have never seen in our PDA approaches. So that's nice that we actually have proof of 25 additional neuropeptides that were never seen before. And for optimization purposes, so if you should want to because now our PRM list only contains 218 neuropeptides. So should we want to extend that to the full neuropeptidome, I think these 69 and these 25 are interesting targets to maybe remeasure the synthetic versions of them and to see if we can add them to this list. But we know that they exist in a biological sample. So it's nice that we have these methods. Now, of course, now this all has been set up, we would like to do some actual differential experiments with it. So our first idea was, the lab of Lisbeth Temmerman is very interested in aging and how the brain actually changes during aging and how neuropeptides are involved in this. So what we wanted to do is, so you see here a typical survival curve of C. elegans. C. elegans has a lifespan of maximal lifespan of about 21, 22 days. So what we want to do was to actually sample over their lifespan. So we've chosen for 3 time points, which is basically, so day 2 adult worms are really young individuals at peak of reproduction. Day 6 are at the end of their reproductive period, so basically menopausal, if you want to extend that to vertebrates terminology. And day 14, we would consider as old individuals. So the plan was to sample across these time points and do a neuropeptide extraction on this and test if we can see differences in neuropeptide levels between these 3 days. So this is what we are currently working on. We do, however, have some trouble with reproducibly sampling these worms. So we're still a bit working on the sample prep side of this and see if we can get this fixed further. But I think there might be some interesting result lying here if we can add our targeted method on this. So then to conclude, I want to thank, of course, my supervisor, Lisbeth Temmerman, and all my colleagues from the lab there, and of course, Christina Ludwig for her help setting up the methods and, of course, running our samples. And then to finish off, if you want to know more about our work, we also have an Instagram page at McWormface. So if you want to see the lighter side of our research, feel free to have a look here. So, thank you.
So thanks.
Thank you for the introduction and thank you for the invitation to present some of my results from topic gathering, liquid chromatography, cannabis business, pesticides, and in which also integrated mass spectrometry. We all know cannabis. It's a very controversial plant. It has usually 2 varieties, medicinal cannabis, also known as marijuana, where THC is known to be the main constituents, and it has quite very interesting pharmacological properties, but it is also incriminated for a series of undesired effects, especially at central nervous system. We all know the high effect that it produces, but also cannabis. We have also the industrial variety, also known as hemp, in which the main constituent is considered to be cannabidiol, also endowed with very interesting pharmacological properties, and it raised interesting recently because it does not have the central nervous system intoxicating effects for which THC is incriminated. The cannabis market is increasing, I would say, exponentially, and is expected to increase. We have very different kind of products, for example, from 3 fields mainly, like personal care and cosmetics, food and beverages, and we can see here we have quite different types of CBD infused foods and beverages, and the pharmaceuticals. In the pharmaceutical, I would highlight the recently approved drug by Food and Drug Administration from the United States called Epidiolex. Is a CBD oil solution used to treat some very rare and severe forms of epilepsy in children that at the moment don't have any other alternative treatment. What we have noticed and how this project started, some of our customers working with hemp products indicated us that there is an issue with pesticides, even though hemp is a robust and fast-growing plant, and no pesticides are normally needed for its cultivation. However, different customers show that when they try to enrich their products in CBD, some of the pesticides are also increasing if they are in concentration, if they are present. And this was mainly, their approach was mainly with conventional preparative techniques. Therefore, we thought about using liquid-liquid chromatography. For those of you who are not familiar with the technique, liquid-liquid chromatography, it is considered a hybrid technique between liquid-liquid extraction, as it uses its high loading capacity, and solid-liquid chromatography, as it has very high selectivity. The setup of LLC is very similar to, for instance, PrEP HPLC. We have a mobile phase and a stationary phase. The main difference is that the stationary phase and the mobile phase are both liquid, and they are practically the two phases of a biphasic solvent system that are obtained simultaneously, as compared to PrEP HPLC, when we first need to select our stationary phase, the column, and then we select our mobile phases. How do we do the setup? We first fill the column with the stationary phase, that is liquid, as I mentioned. We start rotating the column at very high rotation per minute in order to be able to keep the stationary phase trapped within the column due to the centrifugal force. And then we pump our mobile phase, and we wait for column equilibration, which usually means that no stationary phase is lost from the column, and then we collect fractions and analyze them. The chromatogram also looks like a typical chromatogram, but since in liquid-liquid chromatography, one of the most challenging steps is selecting the solvent system, as we have an almost infinite possibilities of solvents that give immiscible systems. So we have some criteria in which to look at. For example, one of them is the partition coefficient, which is practically, for this we need to just analyze the concentration of our target compound into the stationary phase and to divide it to the concentration into the mobile phase. And in order to have to be in a best operating region, so-called sweet spot, we need for this value to be between 0.4 and 2.5. And another parameters is, of course, when we look at two different peaks, we need to have a separate resolution between these peaks, and this is calculated with the help of the selectivity factor alpha, and it is considered that for a good resolution of the peaks, alpha should be higher than 1.5. Our approach in isolating CBD by liquid-liquid chromatography and also removing the pesticides potentially contaminating our starting material is a computer-aided approach that has, in general, 4 steps. We need first to select the pool of solvents, because we have plenty of solvents, but not all the time we can use any of the solvents that we would like to use. We have different regulations. Normally, we ask our customers what are their preferred solvent system or their undesirable solvent system. We group them in different lists, and we try then to make, to create with this preferred solvent system biphasic systems. Then by creating this biphasic solvent system, we try to look for potential solvents that would separate our target compound, in this case, the CBD. How do we do this? We have thermodynamic-based model named COSMO-RS that combines quantum chemistry and statistical thermodynamics. it provides us in the end the activity coefficient that we, with different mathematical formula, we convert it into the partition coefficients. What we need as input for the COSMO-RS is only the chemical structure of our target and impurities. In our case, the target was CBD and the impurities were the pesticides. And in addition, we need the phase composition of the upper and lower phases. This COSMO-RS can also predict this liquid-liquid equilibria, but for more reliable data, we usually use experimental data. There are already, for a lot of solvent system data published in the literature, or in case we don't find and we create a new solvent system, we have GC method to determine these phase compositions. Going back to our CBD, so we screened for a lot of solvent systems. These are just a few examples. Our screening parameter was this partition coefficient p-value of CBD predicted by COSMO. This is, as you see here, the range is a little bit higher than the normal, and we did this in order to compensate for the potential errors of the models. And from COSMO, what it gave us as an idea was that if we want to separate CBD by liquid-liquid chromatography, we need a solvent system to be composed on the one side from water, on the other side for heptane as the opposite polarity, and some bridge solvents, which we've noticed that it can be alcohols with short carbon atoms, methanol, ethanol, acetonitrile, or acetone. Then we went for the shake flask experiments for the CBD within these predicted solvent systems and for the pesticides. And this is where we used LC-MS MS for CBD and most of the pesticides. We downloaded transitions from the literature, multiple reaction monitor transitions from the literature. However, for some pesticides, they were freshly tuned in-house. And then having this data, we also plotted some small calibration, and we extracted for each pesticide the concentration in the upper phase and that in the lower phase in order to calculate our partition coefficient from the experimental point of view. Having all this data, now we ended up with 3 solvent systems to propose the separation of the CBD. And before going to the last step, liquid-liquid separation step, we wanted to see, okay, how was our prediction compared to our experimental data. For this, we worked with a list of pesticides that is regulated in the Oregon state. I have to say that cannabis products are regulated in terms of residual pesticides in different states. For example, in the United States, the number of pesticides is different from state to state, with Oregon and California having the strictest regulations. Canada at the moment has the strictest regulations from all the countries, and European level, since CBD products are quite new on the market. There are discussions, and we are expected soon a similar list of pesticides to also be regulated. We then looked into, okay, how many of these pesticides would interfere with our CBD separation with these 3 solvent systems, and how much of this percentage were predicted already by the COSMO-RS. So if we look at the first solvent system and the highly critical pesticides, I must say that the criticality levels were defined based on the separation factor that I was mentioning, that usually it has to be higher than 1.5. Those for which alpha was lower than 1.5 are considered highly critical and with a low probability to remove them. Then we created 3 other levels, with the last level as the non-critical pesticides. If we look now at the first level, we can see that COSMO-RS predicted a percentage of 8% of the pesticides as highly critical. The same percentage of pesticides were also shown by our shake flask experiments. The similar outcomes were obtained with the third solvent systems, while the second solvent system showed higher percentages of critical pesticides, so indicated us already from this point a lower selectivity for the CBD as compared to the pesticides. However, what I would like to enforce are the non-critical percentages. We can see that Cosmo predicted from up to 50% up to 70% as the pesticides non-critical, and these percentages were lower than those predicted by the experimental, which is good because in this way we compensate for the models, thermodynamic models error. And practically, the main advantage is that already a user which has a list of pesticides that might know that are present in his starting material can already forget about these pesticides, does not need to do shake flask experiments because these pesticides would be completely removable just by having information from Cosmo RS, which also we validated them with the help of shake flask experiments. Having all this data, we propose some guidelines how to select the proper solvent system among the three that we proposed. First of all, we need to characterize our hemp starting material in terms of what pesticides are inside. Our example is with Oregon listed pesticides because we were able to find all these pesticides in a physical format from a provider because it's quite difficult to acquire all the pesticides regulated in each state. And at the moment, the most used methods are based for the analysis of pesticides are LC-MS methods. So having this, we need to see if we are lucky and we don't have pesticides, then we can choose any of the three solvent systems to separate CBD with pretty much the same productivity. Now, the selection among these three solvent system depends on the user preferences. Some might not like acetonitrile because it's more expensive, and for some consider more toxic, and they might like acetone and methanol, or some other users might say that they don't like methanol and they prefer acetonitrile. So this is usually a user dependent choice, and also from Cosmo and from our experience in liquid chromatography, we have seen that many times heptane and hexane are keeping similar outcomes. There is no advantages in liquid chromatography over them. However, hexane is considered by some is considered to have a higher toxicity, but is on the other hand cheaper. So some users might prefer to use hexane instead of heptane, and we had this kind of situations. Now, if we have pesticides, we have to look exactly which pesticides we have. Are these pesticides present on this list here, shortened list? So I must say that Oregon list has 65 pesticides. So we reduce this list to around 12 pesticides. If again, we are lucky and we don't have pesticides, we can use whichever of the three proposed solvent system to separate our CBD. If, however, we have pesticides from this list, again, we have to look which exactly are these pesticides, because we can separate them from the CBD with the use of different solvent systems. For example, for this mixture, we can use the first solvent system. For a different pesticide mix, we can use the second or the third. However, I must say that in each of these solvent systems have their limitations. A total removal from for the pesticides marked with asterisk might not be possible due to the fact that they are on a highly critical zone, and different strategies might be needed, like a second separation with another solvent system or some a complementary preparative technique. Now, we wanted to see what exactly happens if we have a real extract. We have one extract or some extracts from our project partner, hemp extract, but when we analyze it by LC-MS, no pesticides were retrieved within this extract, which was good. But it meant more work for us because we had to artificially spike our extract. And the spiking was performed not with all the Oregon listed pesticides. We just selected some of the most critical that we have seen in the previous guidelines. And we did liquid chromatographic experiments on a small scale unit that has a column volume of 18.2 milliliters, just for the proof of concept to see what happens. And for example, having this pesticide spiked matrix, we've seen that with the first solvent system, most of the CBD fractions were quite pure. Containing only the beginning and at the end of the CBD peak, there were some traces of pesticides. However, with the same matrix in the second solvent system, more than 50% of the CBD collected fractions contain some pesticides. Even if for this matrix, pesticide matrix, the outcomes with this solvent system were not quite good, we have seen that there are different selectivities for other pesticides that this solvent system, for which this solvent system has advantages. And the best outcomes in our case we obtain with the third solvent system. As you can see, most of the CBD collected peaks were lacking the pesticides. To conclude, I would like to say that our computer-aided approach is a valuable tool to screen and select the biphasic solvent system. In our case, for the simultaneous CBD isolation and pesticides removal with liquid chromatography. But the proposed approach can be used in general and can be applied to other matrices, other target components and impurities, can be from any natural matrix or can be synthetic matrix, as long as we know what our target are in this case, and what our impurities or undesired compounds are, and we want to remove them. More details are available in our recently published paper in Industrial Crops and Products. I would like to thank Karin and BayBioMS for the support in conducting the LC-MS analysis. Also, thanks to my colleague from our department and our funding agency, BF, and our project partner, BAFA-NOI. Thank you very much for your attention, and I'm open for questions, and for further information, please don't hesitate to contact me.